Hello, everybody. Welcome to another talk in Lasmo Week. And I hope all of you guys are taking care and are safe during these really difficult pandemics that we are going through. And before we start, I would like to introduce myself and tell you guys a little bit about myself and why I got involved with biology, okay? And so, first of all, my name is João Paulo Capretz Batista da Silva, and currently I am an associate professor at the Federal University of Paraíba in the city of João Pessoa, the state of Paraíba, in the northeast part of Brazil. And so I live uh, in a city that is at the coast, northeast coast of Brazil. So we have beautiful beaches here, and eventually I can go and interact with the group of fishes that I work with, which includes the chondrichthyans, the sharks, skates, rays, and chimeras. Okay, so right now I would like to show you to start my talk and show you guys a little bit about my the times when I was a kid and a teenager, and that I really loved to interact with museums and see every kind of animals, the huge diversity that we have, especially here in Brazil, South America, and it is amazing. Okay, so just to start, uh, and putting here a laser point just to make things easier. Uh, Here I am when I was a little boy, and guys, I can tell you that I love to go to a museum in a city that I grew up that is called Campo Grande in the state of Mato Grosso do Sul. So this city in particular was really close to the wetlands of Pantanal. So just like as you know in the Amazon region, Pantanal has a huge diversity of animals. Not only animals but also plants. But my interest was from the beginning uh, directed to the animals. I do like them. And so as you can see here, it is myself with my two sisters. They like to visit this museum here too in Campo Grande that is called the Indian Museum. So this museum had every kind of artifact that was related to the Indian culture, but also they have taxidermized animals, as you can see here. They were uh, already dead animals, and they were prepared for an exhibition. Just as like you have uh, several beautiful and amazing museums in the US. Okay, so just to illustrate, we have here a tuyuyu, which is a bird that is characteristic from Pantanal, and it has that dark head with a red collar here, and most of the body is white. It has long legs. We have this museum had a huge diversity of reptiles, including some huge snakes, just like a sucuri, and uh, the caiman or alligators, turtles. Of course, this is a marine turtle, but they had this animal in their exhibition or exposition. And also, they had a beautiful entomological collection, and as you can see here, beautiful butterflies with this vivid blue color, and also this group of butterflies here, and they had primates, armadillos, and what else? We have an endemic species of wolf in South America that we call lobo-guará, and they also had this animal in their exhibition. A feline, the jaguatirica, which resembles a small uh, jaguar, a tapir, which is a huge mammal, and it's a beautiful animal. But of course, they also had fishes, which is my group of study and which I really like. So here we can see some piranhas that they have highly developed teeth that they use to tear apart flesh, okay? And another beautiful fish that we have in the Amazon region, which is called arapaima, which is a really big fish and it has a really good meat as well. So when I was young, I was amazed and I always asked my mom and my dad to bring me to this museum. It was my favorite part of the weekend. And every time that I had the chance to go to this museum, I was just like really, really happy, amazed. It was a wonderful place for me. So as you can see here, I was astonished by all the beauties and the huge diversity of animals that I was seeing at the exhibition, and I can tell you that I was really, really happy. Later on, when I was a teenager, when I got the chance, I would ask my mom or my dad to travel and go fishing. So I was really fortunate to have this kind of experience. So uh, at one point in my life, I had to move far from Campo Grande, Mato Grosso do Sul, which was close to Pantanal, and I moved to the countryside part of Brazil. So I was really far from Campo Grande and the wetlands of Pantanal. But in 2000, more specifically, I had the chance to go back there and go fishing with my parents and my sisters. And in this occasion, when I went out fishing with my dad, as you can see here in both of these photographs, I had the chance to fish freshwater stingrays. And I didn't know that when I was an undergraduate student that I would start working with this group of fishes. And it was really, really nice. It was a great experience again. And as you can see, I graduated in biology. That was my choice, of course, because I was in love with animals since I was a kid, especially with fishes. And my choice was not that difficult. So I graduated and I was really happy, of course, because finally I was a biologist. Okay. And just showing a little bit more information about myself and my academic background. So I graduated at the University of São Paulo in 2006, and I started working, as I said before, with freshwater stingrays, the taxonomy of this group. And to start, I just explored this species, Potamotrigon falconeri. During my master's, I moved from Ribeirão Preto, where I graduated, that is in the state of São Paulo, and I moved to the capital, São Paulo, the city of São Paulo. And I went to another institute uh, that is related to the University of São Paulo, which is called Biosciences Institute or Instituto de Biociências, and I did my master's there working with another species of freshwater stingray, which is Potamotrigon urbigine. And I stayed in São Paulo to do my PhD, and I thought that it would be a good idea to change a little bit the subject of study. So I left behind a little bit the freshwater stingrays and I started working with the class chondrichthyes as a whole. And I studied involving the comparative anatomy of the appendicular skeleton across this beautiful class of fishes. And as a result, I could propose a phylogeny for the group based only on morphological characters. And finally, during my postdoc, abandoned completely the constrictions. I just say abandoned, but I still work with the group. I love this group of fishes, but I thought that would be a nice experience to do a comparative study with a group of bunny fishes, which are the elopomorphs. So I worked with them. And for those of you that don't know uh, what I'm talking about or what group of fishes is this, elopomorphs include the tarpon and several beautiful species of eels. And I think uh, the most conspicuous one is are the moray eels. Okay. So I finished my postdoc in 2019 in September, and in November I was hired as an associate professor at the Federal University of Paraíba at the northeast part of Brazil, which is where I'm currently working, teaching and developing my researches. Okay, so João Pessoa in the state of Paraíba is a really beautiful city, coastal city. As you can see here, we have beautiful coral reefs, which are really close to the shore, as you can see, and we have a huge diversity of fishes associated to this coral reef. And if I'm not wrong, in 2017 we had a meeting, a really nice meeting here. And that was called Sharks International, and I hope it can happen again very soon. Okay, so right now we are gonna start talking a little bit about the neotropical freshwater stingrays, which is a really nice group of fishes that is restricted to the South America. And we are gonna see what are the characteristics of the group, the systematics, and a little bit of the works that have been done exploring their interrelationships. Okay, so just to start, I would like you guys to know what are the characteristics that identify or describe a chondrichthyon. So every chondrichthyon they have or a fish that belongs to the class Chondrichthyes. It has a cartilaginous skeleton, but it's not the most interesting part of the skeleton of these fishes. Their cartilaginous skeleton is covered by a different kind of mineralization that we call perichondral prismatic calcification, forming this beautiful honeycombing pattern that, as you can see here, at the lower jaw of this ray, a neurotrigon. Okay, so we have a different kind of mineralization that is restricted to the periphery of the cartilage, forming this beautiful honeycombing pattern, as you can see here. Okay, so the chondrichthyes they don't have bones, endochondral bones, as we do as mammals. All of us we have an endochondral bone. We have bones. These fishes they don't have bones, and their dermal skeleton is formed only by placoid scales or dermal denticles, as we are gonna see um, a little bit after during this presentation. So the males of Chondrichthyes, they have claspers, which they employ to transfer sperm to the female. So the male introduces a clasper in the cloaca of a female, and during copulation, a kind of behavior that we also see in mammals. Okay, we know that every mammal has a penis that he introduces um, in the vagina of a female mammal to during copulation. So the same kind of thing happens with chondrichthyes. Okay. Another interesting characteristic of the group is that they replace their teeth continuously. So you're not gonna see any shark ray or chimera without a teeth, and this is really cool. And they have a huge diversity or morphological plasticity when we are talking about teeth. And another interesting characteristic of the group is the presence of the placoid scales or dermal denticles or scales only, and each that is a structure from the dermal skeleton that is related to the reduction of drag during swimming. Okay, so another thing that is really cool about the chondrichthyes is that they have highly developed sensory organs, just like ampullae of Lorenzini, which are uh, uh, special structures that they can feel or sense electric stimuli, just like when a fish is swimming, the electric impulses that are forming during the swimming activity can be sensed or felt by any chondrichthyon, okay? Another thing that's really cool about these fishes that they share with other groups of fishes is the presence of lateral line canals, which can sense also movements of water, so they can sense if there is a predator or a prey nearby, okay, just during um, the movements of water. And they have special cells inside these canals that we call neuromasts, okay, and they can feel the displacement of water throughout this, the interior of these canals. Okay, so these are sensory organs that are found within this group. They are really or highly developed and specialized. So we're gonna try to understand what now where are the group of freshwater stingrays placed and why they are part of a huge group that we call elasmobranchi. So elasmobranchi includes the sharks and rays, and within elasmobranchs we find a superorder of fishes that we call Bathoidea, which includes all the groups of rays. So there is a debate nowadays about the monopoly or the validity of several groups of rays, but we are not discussing about this right now. The only thing that I want you guys to understand is that every all the groups of rays they share specializations or body modifications. Okay, so the first one is related to the formation of the disc. And what is the disc? We can see highly developed pectoral fins, which we don't see in other group of fishes, just like sharks. And this pectoral fin is connected to the head, forming that structure that we call a disc. So at the center or at the middle of the body of a ray, we can find the head and and the belly and the viscera just like inside here. And at the laterals, we have most of the body formed by the pectoral fins. And when we try to understand the skeleton of a ray, we can see that the pectoral fin is connected to the neurocranium or the cranium of a ray, which is a really cool characteristic of the group because we don't see this kind of modification in any other group of fishes. Okay. Another thing that is only found in the group of rays is that all the branchial slits they are ventral. So when we try to analyze a shark at the left side, uh, at the bottom left side here, we can see that the branchial slits they are lateral. And when we try to see where the branchial slits are in a ray, they're always gonna be at the ventral portion of the body. Okay. Another cool characteristic about elasmobranchs is that all the members of this group they have modifications of the vertebral column, and one of these modifications we call synarqual, which is a structure formed by the fusion of vertebrae. Okay, so we have the combination of different vertebrae to form this compact structure, which is discrete and single, and which we call synarqual. And trying to understand a little bit about the diversity of the group of the bathoids or race, we usually identify five different orders. The first one is the order Torpediformes, which includes the electric race, and the other one is the order Pristiformes, which includes the sawfishes, which is a ray that has this really elongated rostrum with lateral teeth, which we call a saw. Okay, the order Ragiformes, which we usually call the skates, the order Rhinobatiforms. So when we try to understand the morphology of the body of this guy here, it resembles a guitar, so that's why they are called guitar fishes. And finally, the group that we are gonna see uh, more information about today. It's the order Mediobatiforms that includes all the rays that have a caudal instinct that they use for defense mainly. Okay, and here we have a beautiful species which is Aetobatus narinari. Here we call this ray Cheetah ray because its color pattern resembles a little bit the color pattern that we see in this feline, which is the cheetah. Okay, and when this group of rays they are swimming, they have a little beautiful kind of swimming that we call oscillatory. So it looks like they are flying under the water, and it's really beautiful. And all the Emiliobatiforms, as I told you before, they have a caudal sting at a portion of the tail. Usually it is located at the distal part of the tail, but in several groups you can find this caudal sting at the base, and it has serrae, lateral serrae, and in some cases there is also an epithelium that can produce venom. Okay, so as you can see here, the caudal sting can be positioned or located more close to the end of the tail. It can be at the mid part or mid portion of the tail in some cases, and in another cases it may be located more close to the base of the tail. Okay, and as I told you before, the caudal sting is usually covered by an integumentary sheath, and this integumentary sheath is the kind of tissue that it is responsible for the production of venom that we see within this group. So every mediobite form, as I told you before, has a caudal sting, and when we try to understand the structure of this sting, we see that it has a long axis, and laterally we find several serrae. Okay, so we usually say that this sting is serrated, a serrated caudal sting. So the core of this sting is made of dentine and is covered by enamel. So the basic structure of a caudal sting or of every placoid scale is really similar to what we see in our own teeth. And this structure, this sting, it is covered by an integumentary sheath which is formed by a specialized epithelium that has the capacity or it's able to produce venom. Okay, and just to illustrate here what I'm trying to say, When we try to understand the morphology or histology of the caudal sting of a freshwater stingray, as we can see here in a Potamotrigon falconeri, and we see a cross section of the caudal sting, we can identify here at the core or the center the caudal sting, and covering all the mineralized portion of the sting, we see a peripheral epithelium or integumentary sheath that has several modified cells that can produce a really potent venom. Okay, and one really nice thing about freshwater stingrays is that according to the species that we are studying, we can see several morphological characteristics that can be attributed to different species. So here we're gonna see a little bit about this guy here, which is an Eliotrigon, but we can see that its spine don't have that developed serrae as we saw just before, and as we can see in other species or genus within the subfamily Potamotrigonini. Okay, so the morphology varies according to the species, and this is a really cool and important kind of information because we can use it as a taxonomic tool, for example. And when any mediobite form, especially some marine species as Dasyates or freshwater stingrays more specifically, they are disturbed or pulped, and so eventually we can step in a freshwater stingray, and they use that sting to protect themselves. And the wound is just like severe because, as I told you before, they have that epithelium that can produce venom, but they are not aggressive animals. Okay, they are really they are calm, and they just have this kind of negative interaction with people because accidentally we can step on them and they think they are being just like stroke or they are being attacked by any kind of animal. So that is a defensive posture, and any kind of organism would have the same reaction. So they are not. What I'm trying to say is that they are not aggressive animals. Okay. And another thing that is really cool about forms is that besides having the synarchal, they also have another fusion of vertebrae that we call a second synarch and this is found only in rays of the order Myliobatiformes. So here we can see the skeleton of a Potamotrigon motor, which has a really beautiful uh, pectoral fin skeleton, as you can see here. So we can see the pectoral basals, the propterygium, meso and metapterygium in several highly developed radials that support the fins. Okay, and we have a beautiful and huge diversity within Myliobatiformes. I showed you before the eagle ray that we call here in Brazil the Cheetah ray. We have several species within the genus Dasyates. More recently, a family, Dasyaridae, suffered a huge rearrangement, a taxonomic rearrangement, but nevertheless, the diversity of the group it is the same. It's just like we understand the group that was modified. Um, we have here another beautiful species, Taniura lima, which has this beautiful round and vivid blue round spots over the dorsal portion of the body. The manta ray or devil ray or eagle or devil ray, sorry, which are filter feeding rays that also have this oscillatory kind of swimming, and the butterfly ray, which has highly developed pectoral fins, as you can see here. And it's difficult to say now when we see the morphology of the body of these fishes that they are rays or a fish because it's really modified, but they are really beautiful animals. So right now we're going to finally start talking about the family Potamotrygonidae that was firstly proposed by Garman in 1877. And before, the family Potamotrygonidae only included neotropical freshwater stingrays, but it was recently rearranged in 2016, and now it includes also two members uh, of marine species. Okay, and members of the marine species, they are part of the subfamily Styracorini, uh, which includes rays within the genus Styracura, as you can see here, Styracura shimardae. And the subfamily Potamotrigonini only includes freshwater stingrays. So within this subfamily we have four genus, sorry, Eleotrigon, Paratrigon, Plesiotrigon, and Potamotrigon. Okay, we're gonna see the diversity within these genus or genera of rays in the following slides. And the distribution of this group of species, they are restricted to the freshwater environment, uh, is is endemic or exclusive to the South America. So as we can see here, we have members of the genus Eleotrigon, Paratrigon, Plesiotrigon, and Potamotrigon distributed all over the South America. This map here is not complete. I'm gonna show you another map that has a more updated data on the distribution of the species. But what I can tell you is that we can find representatives of the subfamily Potamotrigonini uh, in all the hydrographic basins or watersheds across the South America, except those that drain the Atlantic first and that flows to the Atlantic Ocean at the coast part of Brazil. So at this portion, we will not find any species of Potamotrigonini. Okay, so this is the updated map that we have for members of the subfamily Potamotrigonini. So they are distributed in most part of South America. Of course, they are not found in all of the countries of South America. But when we talk about Brazil, we can see that the rivers that drain the Atlantic first from the northeast to the south part of Brazil, we will not find any species of Potamotrigonini, okay? But they are really diverse in the Amazon region and in parts of the Paraná-Paraguay basins, hydrographic basins, okay? And members of the subfamily Potamotrigonini, they also have some exclusive characteristics that we call synapomorphies, which are found only in the species, but they are not found in any other group of fishes. So the first of them is what we call a developed prepelvic process at the pelvic girdle. So this is a site for the attachment of the uh, ventral portion or ventral bundle of muscles. So it gives a huge support to the pelvic girdle. And this is important because when we try to understand the behavior of this group of fishes, we see that they are associated to the bottom of the rivers, okay? and they use the pelvic fins for locomotion as well. Not only they don't use only the pectoral fins. So when they are at the bottom of the rivers, they can use the pelvic fins to for locomotion. They also have what we call angular cartilages that give support, and they are involved in the suspension of the jaws, okay? And the angular cartilages are these small cartilages, as you can see here. They are in between two skeletal elements of the jaws. The first is the mandibular, and the other one is the Meckel's cartilage. Okay, so their function is only related to the suspension of the jaws to give support to the jaws. Okay. And another important characteristic of the group is that all freshwater stingray they have the rectal gland reduced. And what do I mean by that? We know that every marine elasmobranch sharks and rays they have developed rectal glands that is involved in the extraction of the salt from the bloodstream. So these animals they drink salt water, so they have to eliminate the excess of salt from the bloodstream through a special organ that we call the rectal gland. So as freshwater stingrays they are restricted to the freshwater environment, they don't need to have a developed rectal gland. Okay, so theirs is really reduced. And now we are gonna try to understand a little bit of the diversity of the group. So we can find four genera within the subfamily Potamotrigonini. Okay, so we have Eleotrigon, which has two valid species, Paratrigon with three valid species, Plesiotrigon with two valid species, and Potamotrigon with 31 species. So you can see that the genus Potamotrigon is the most diverse that we have within the subfamily. And here just showing you a little bit about the diversity within the genus Paratrigon. So we have Paratrigon aereba, and uh, recently in 2021, this year, we have the description of two new species, Paratrigon orinocensis and Paratrigon parvaspina, by a really close friend of mine, Thiago Loboda, and these are really beautiful animals. They are also known as apple rays because the shape of the body, the morphology of the body resembles the shape of an apple. Okay, so it has a faint notch at the anterior portion of the disc, which resembles what we see in an apple. That's why they are called here in Brazil apple rays. The other genus that we have within the subfamily Potamotrigonini is Eleotrigon. They have this really round body, as you can see here. So if you want to do a pizza for your friends, you just have to buy some tomato sauce, cheese, and you put the tomato sauce here, cover with cheese, put it right in the oven, and you have a really nice pizza. Just kidding, as we can see, this ray here resembles a lot the body morphology of Paratrigon, as we saw in the previous slide. And they are really huge animals. For instance, members of Paratrigon and Eleotrigon, they can reach more than 1.5 meters in width, and they can reach to 40 or 45 kilos of weight. So they are huge animals, they are huge fishes. So the other genus that we have within the subfamily Potamotrigonini is the genus Plesiotrigon. So these are really beautiful rays as well, and they have a really elongated tail, as you can see, resembling a whip. and their eyes are small, as we can see in Paratrigon and Endotrigon, but their body morphology in general resembles what we're gonna see for the other genus in the last one, which is Potamotrigon. So when we enter Potamotrigon, we enter a world of wonders because this is the most spacious genus within the subfamily Potamotrigonini. So we have 31 species, describe the species for this genus, and we can see really colorful and different color patterns over the dorsal portion of the disc in this race. So, for instance, in Potamotrigon leopoldi, we have this dark or black background with several white spots that are rounded and they are larger than the diameter of the eye. And we have Potamotrigon wallacei, which is known in Brazil as Potamotrigon cururu, which is a really small freshwater stingray. That would be a, a dwarf Potamotrigon. Okay, we have Potamotrigon schroderi that has this beautiful color pattern here. We're sampling that would be just like a Rosiris, or it is a highly convoluted uh, vermiform formation that we see for these spots here over the dorsal portion of the disc. We have also other beautiful species within the genus Potamotrigon, as we can see here, just like Potamotrigon germani, Potamotrigon lemai, that have these different color patterns. And the one I like the most is Potamotrigon jabuti. Jabuti is a popular name that we use to call several species of turtles in Brazil. And specifically this ray here, it has a color pattern that resembles the shell of this turtle. So when we have the chance to collect these animals in nature and see the really variable color patterns that we can find for them, it is amazing. That's what I can tell you. And we have, for instance, Potamotrigon brachyuria, which can reach up to 2 meters of disc width, and they can reach up to 200 kilograms. So they they are really huge freshwater stingrays, and of course that the animals that are really huge, they are older in age, but so far species exemplars or representatives or members of this species Potamotrigon brachyura are the ones that we have records for the largest disc width, or the largest, the highest values for the weight uh, for the genus as a whole, or also for the subfamily Potamotrigonini. Okay, so I'm just trying to illustrate the huge variable color patterns that we have for these freshwater stingrays. They are really amazing, and because of that, we usually have people that like fish keeping, and they are explored for this purpose as well. Okay. And a cool thing is that we also find parasites inside the body of freshwater stingrays. They are animals that we studied that have a huge distribution in South America, and there is a group of researchers studying the parasites they can find within the body of these animals. And it is really interesting because we know that the story, the evolution of this group of rays is totally associated to their parasites. So uh, there is a there are some methods to study these parasites. So they are found in the gills of freshwater stingrays and also in their intestine. So we know that elasmobranchs, they have a modified intestine which forms this structure that we call a spherical valve, okay? And when we dissect the intestine of a freshwater stingray or its gills, we can find these worms here, these helminths. And we know there that the story of the evolution of this group of worms is totally associated to the evolution uh, of this group of fishes, which are the freshwater stingrays. And just to illustrate here how ugly these worms are, uh, they are really diverse as well. As we can see, the diversity that we see for the freshwater stingrays within the subfamily Potamotrigonini is huge, but the same can be said about their parasites. So they are not beautiful as the rays are. They have this bizarre morphology, but they have been studied for many years by a group of researchers in Brazil, but not only Brazil, across, outside Brazil as well. And it is also a really interesting group to be studied. Okay, and continuing with our uh, cool group of fishes, the family Potamotrigonidae has or presents huge taxonomic problems, and this is related to the huge polychromatism that we see for this group of fishes. And when I say a polychromatism, what am I talking about? So there is an intense interspecific variation in coloration. So when we are exploring or analyzing specimens from the same species, we can find really different color patterns. And at the same time, we can find similar color patterns in distinct species. So when we see these figures here, it's hard to understand what I'm trying to tell you guys. But for instance, in remembering you, the color pattern that we can see for the dorsal part of the body of these animals is really variable. And how can we tell species apart here? Look at this huge color variation that we see within the subfamily Potamotrigonini. So it's a really difficult uh, job for us to try to understand this uh, color variation and to tell accurately uh, which species is each. So it's difficult for us to say uh, if a different color pattern belongs to a species A or a species B, for example. So just to illustrate to you guys, there are the coloration also leads to the proposition of names of uh, uncertain validity. But just doing a simple exercise right now, how many species do you think are there here? There is only one species, there are two species, four species, and so on. So analyzing only the color pattern is a really difficult task to tell apart species within Potamotrigon. So I can tell you with confidence, because I have studied this group of fishes, that when we analyze these six different color patterns, we can identify three different species. So we have here Potamotrigon urbigine, Potamotrigon humerosa, and Potamotrigon marini. Okay, and to make things more difficult, we know there is also an ontogenetic change of the color pattern uh, throughout the life of several species. So when we analyze a juvenile or a young member of a species, its color pattern is not similar to what we see in an adult. So we have to be really careful to not identify or propose new species when we are analyzing young individuals, okay? And to try to help in this task, we usually employ the morphology of the dermal denticles. So the dermal denticles, they are a really important tool for the identification of several species within Potamotrigo, for example. And why is that? When we see the morphology of dermal denticles, we understand that they vary according to the species. Look at the morphology of the dermal denticle of Elytrigon, for example, and compare it to Pontamotrigon motoro. So here it resembles a snowflake, and here it looks like the the foot of a cow. So they are not similar. So we can employ the different morphology of the dermal denticles as a taxonomic character. Okay. Another cool thing about freshwater stingrays is that their dentition is really variable and it can also be employed as a taxonomic character. For example, we can find sexual dimorphism um, for males and females. Males, they usually have a triangular cuspid on their teeth, which we do not find in females. And another interesting thing is that the dentition of the species within Potamotrigonini, they vary across species. So it reflects different kind of feeding options that they have or feeding behaviors. For example, Potamotrigon urbigine, which has these three cuspidate teeth with three triangular projections here, it is specialized for feeding on insects. Potamotrigon motoro, for instance, it has lateral enlarged teeth, forming these placoid teeth as we can see here. And we know these animals, they feed primarily on crustaceans or mussels, okay? And this is really interesting. Another important aspect is that according to the feeding options that a species exhibit, it is reflected on its feeding behavior. So we can see here that Potamotrigon motoro, for instance, will scan across the river bottom to see if it can find a small crustacean or a snail somewhere, and it will crush that animal and its hard shell with its modified teething plates, and Potamotrigon urbigine or Potamotrigon falkneri, more precisely, that feeds on fish, will just like trying to trap other species of fishes to feed on them. Okay? Another cool thing that we can find for the freshwater stingrays is their ventral-lateral line canals. As we can see here, when we dissect the ventral part of the body of any freshwater stingray or any ray and remove its epidermis, we can see the arrangement of the ventral-lateral line canals. And these canals, they are developed ventrally because the body of these fishes, they are flattened dorsoventrally, okay? And one cool thing is that when we try to understand and analyze the variety of the ventrolateral line canals, we know that they are different across the different species that we have for freshwater stingrays. Just to illustrate here, we have different species and of freshwater stingrays. Most of them are of the genus Potamotrigon, but we have here a Paratrigon as well. And when we uh, can see their arrangement and morphology, we know that they are really variable across the species. And one important thing is that the verbal arrangement of these canons they can be employed as taxonomic characters and also as phylogenetic characters. Okay. And another important thing is that in Paratrigon we can see that these canons they are more complex and highly branched anteriorly. So this may reflect the preference of habitat of this species that is always found at the bottom of the rivers, but the species of Potamotrigon they may swing across the water column, okay, which is not a common thing for Paratrigon, for example. Another important character that we usually study for freshwater stingrays is their skeleton, which is, as a general, is conservative, but we can find some differences that can be employed not only as taxonomic characters, but also as phylogenetic characters, okay? But here just to illustrate how beautiful the skeleton of this species they are, and as any member of the superorder Batoidea, they have highly developed tactile fins, and this is reflected here by their highly developed pectoral fin skeletons. Okay, and another important thing, as I told you at the beginning of our talk today, is that males they usually have or they always have a clasper. Okay, which is an intromittent organ. And the clasper also has an internal skeleton for its support. A cool thing that we know about the origin and diversification of freshwater stingrays is that they probably originated from a marine ancestor that invaded the freshwater environment of the South American continent during some events that we had in the Miocene from 23 to 10 million years ago that we call epicontinental seas. So we have the sea invading the continent, and in one of these occasions, this marine ancestor just got trapped in South America and subsequently was responsible for the origin and diversification of the four genus or genera that we saw for the subfamily Potamotrigonini. Okay, and uh, we have a recent work from Fontenelle and collaborators just exploring all these events of diversification of the South American or neotropical freshwater stingrays from their origin through an epicontinental sea and their diversification across South America is closely related to the formation of the Andes Cordillera, okay, to this chain of mountains at the west part of the South American continent. Another interesting thing related to the origin of the freshwater stingrays is that the same ancestor marine population that was distributed on the northern portion of South America and that was part of this population was trapped during the Miocene through epicontinental seas in the continent. The same ancestral marine population went through a different Vicarian event and gave rise and or originated two different species that we call today Styracoda chimardi and Styracoda pacifica. and which are closely related to members of the subfamily Potamotrigonini. Okay, so we know that during the formation of the Panama Isthmus, we had the appearance at east of the species Styracoura shimardi, which is distributed over here of the Caribbean Sea and the east portion of the Central America. And at the west portion of Central America, we had the appearance of the species Styracoura pacifica. Okay, so the species within the subfamily Styracurini, they are so closely related to the freshwater, neotropical freshwater species of the subfamily Potamotrigonini that they were recently rearranged and included in the family Potamotrigonidae. Okay, so as I told you before at the start of my talk, both subfamilies are within the family Potamotrigonidae. But before, as I told you, only Potamotrigonini was only members of the subfamily Potamotrigonini were considered Potamotrigonides. Okay, so we can see here the phylogenetic position of the species of Styracura. They are not that are now allocated at the genus Styracura, but were before at the genus Imantura. In the we can see that they form a sister group to all of the other neotropical freshwater stingrays of the subfamily Potamotrigonini. Okay, and in this very year, we had the proposition of a new systematic study, phylogenetic study from Fontenelle and collaborators, in which they establish new interrelationships within the subfamily Potamotrigonini. And right now, I am conducting with other collaborators a phylogenetic study to understand the interrelationships of members of the subfamily Potamotrigonini, but instead of using molecular data, as Fontenelle did, exploring nuclear and mitochondrial DNA, we are trying to understand the morphology of this group to propose phylogenetic interrelationships for its members. Okay, and what we do know right now is that uh, members of Styracura are closely related to Potamotrigoninis, and Potamotrigon and Plesiotrigon are sister groups, and they form a collateral sister group to Paratrigon and Elatrigon. Okay, and we have been doing several taxonomic and revisionary works, and we have been raising a consistent and large number of morphological characters to understand the interrelationships of this group of species of freshwater fishes. And so far, we have raised 94 characters, and many of them corresponding to characters related to coloration, spines, or placoid scales, or dermal denticles, ventral lateral light canals, to the skeleton, to the musculature, to the encephalon and dentition. And we were able to propose a really cool tree uh, showing the phylogenetic relationships within the subfamily Potamotrigonini, and we hope that we can publish the results of this study, uh, if not next year, in 2023, because we are doing a huge morphological work. And you know, guys, it's not an easy task to dissect and explore the morphology of any group of fishes, okay? And these are my collaborators, Tiago Loboda, which was at the Federal University of Acre in Rio Branco, state of Acre. and my colleague here from my department, Professor Ricardo de Souza Rosa, that is a professor at the Federal University of Paraíba here in João Pessoa, at the state of Paraíba. Okay? And having said all these beautiful things about freshwater stingrays, I thank you all guys, and I'd like to thank all the support that I have received to conduct my research from several uh, institutions. Okay, and also I'd like to thank my students, Jade, Marcos, Henrique, my colleague and professor Manuela Marinho. We have two cool pages of fishes at Instagram. The first one is Tubarões na Praia or Sharks on the Beach. And we post several nice things about sharks and rays there. And we also have a page which is called Peixes UFPB or Fishes UFPB, in which we post several activities related to our laboratories. Okay. And finally, I would like to thank my really good friend and extraordinary wife. She's an amazing person. My mom, my father, my sisters, Diego Vaz from Harvard, Thiago Loboda, Alessio da Tovo from Museu de Zoologia da USP, and André Casas. All these people, they were really important for my career, and I will be always grateful. Okay? Thank you very much, guys. I hope you stay safe. Take care. See you.